speaker, Dr. Jim Nestler. He attended graduate school at the University of Colorado and has spent the past 26 years, yes, 26, as a professor of biology at Walla University. Some fun facts about him is that he loves, can you guys hear me? That he loves scuba diving, hiking, backpacking, loves dogs. He is terrified of heights, but loves bungee jumping. He is scared of the dark, but loves caving. And this was the most interesting fact of all. In his next life, he wants to become a maple bar donut. A maple bar donut. So if you want him to do anything for you, just get him a box, just get him a dozen maple bar donuts, and he'll do it for you. So without giving much away, he's going to be talking about zombies of the sea. We're very grateful to have him here with us and for taking time from his very busy schedule to give this talk. So without further ado, I introduce Dr. Jim Nestler.
Thank you very much. Maple bars. My students give me maple bars, they get A's. And is that the same way here too? Yeah, probably not. It's great to be here at Southern. It's a wonderful opportunity to come visit a different campus, see my fellow biology professors, see and talk to a bunch of wonderful students. I think I talked to about 10 students today. Thank you, each and every single one of you. Sea cucumbers, rotting and regenerating zombies of the sea. What in the world is all that about? Well, before we talk about those, let me briefly talk about this coming summer. Some of you may be aware, and some of you may not be aware, that there is a Seventh-day Adventist Marine Laboratory on the West Coast. It's owned by Walla University, but every summer we have students from Southern, Andrews, Union, La Sierra taking a variety of different classes, and so I thought I'd quickly share this with you. I think many or most or all of you are either in Gen Bio or have taken it already, but if you have friends who might need it, we offer the full school year's worth of Gen Bio up at Rosario during the summer session. And we also offer a variety of upper division classes you'll see here on the right. We have a marine biology class, a marine phycology class, which are algae, and I met someone today who loves algae, a class called Marine Environmental Physiology, and then one called Sea to Summit. You'll see that two of the classes up there are kind of a slightly different color. That's because they'll be taught by Southern teachers. Joyce Acevedo will be teaching general biology, and Ben Thornton will be teaching the upper division Sea to Summit. So if these are things that you're interested in, feel free to talk to me, talk to one of those teachers, or you can talk to Keith Snyder, because they know a lot about this particular program. Now, there are some things that you need to know about me in addition to my love of donuts, and let me share some of those with you. First of all, even though I've been doing this job for 26 years, sometimes I feel like I still don't know what I want to be when I grow up, or if. And secondly, I really like doing puzzles, but sometimes I don't have the patience, and sometimes I need a lot of help to complete jigsaw puzzles. Now, what in the world do I mean by these two things? First of all, I still don't know what I want to be when I grow up. Some people will ask me, well, what kind of biologist are you? And I'm not really sure how to answer that, because I don't know what kind of biologist I am or what I want to be. You know, there are lots of fields of biology. And if we look, for example, at the National Science Foundation's website, we can see a great variety of different types of biology. For example, they have a thing called population and community ecology. I do that. I study marine organisms in their natural environment. I look at how they interact with each other. I'm an ecologist. But you know, I also like studying the individual animal and what it does on a day-to-day basis, on an hour-to-hour basis. I like studying behavior. Maybe I'm a behavioral biologist. I love studying how different organ systems develop, how you go from a single cell to now a full organ. Well, I guess I must be a developmental biologist. My favorite, though, is physiology. Oh, respiratory systems, cardiac systems, endocrine systems are just awesome. I guess I'm a physiologist. But I also do a lot of cellular biology, what's happening on the cellular level, on the molecular levels. Maybe I'm a cell and molecular biologist. So I don't know what I am, But I know that in order to study a particular organism, even a small part of that organism, you really need to know how that organism interacts, not only within itself, but with the entire environment around it. And so in all of my research, I have a variety of students who are helping me. Some want to study ecology, some want to study molecular biology, and it's awesome because I love them all. Puzzles, I like puzzles. And for me, science is a puzzle. And tonight we're going to be looking at how we're slowly solving the puzzle involving this particular species, which we'll look at a lot later, called Parasticopus californicus, a sea cucumber. There are some things we know about this particular animal. There are many things we don't know. And I can't solve this puzzle by myself. I'm going to need a lot of help. So what's this whole thing about these sea cucumbers and they're rotting away and they're regenerating? Well, let's first talk about what a sea cucumber is. Many of you probably know, some of you might have forgotten, that these are animals within the phylum Echinodermata, the echinoderms. We're used to sea stars and urchins and brittle stars. And we know that they have this pattern of five rays and five this and five that. Well, sea cucumbers are merely these five-rayed organisms where the rays have come together now to form basically a long tube. So they have five strips of muscle. They have 5 rows of tube feet. They are echinoderms. But they're in the class called Holothuroidea, so we'll also hear about holothuroids or holothurians. I call them cukes because it's a nice short way to talk about cucumbers. There are a variety of cucumbers. Some are as small as your little finger, like this white one here on the left. Some of them can be up to almost 6 feet in length. The largest sea cucumber, Thelonota ananas, that we see here on the bottom. Some of them are filter feeders, like the one that we can see on the top, put out its tentacles. The animal doesn't move at all. Other ones will crawl across, sucking up muck, basically acting like vacuum cleaners. Well, this is what the outside looks like. Let's look briefly at the internal anatomy of a generic cucumber. This is a tropical one that we're looking here. And if we look at the very top, you'll see these sort of black structures here. These are the feeding appendages, or they're called the tentacles. That's where the mouth is. Well, so you got the mouth at one end, and if you got the mouth at one end, well, you're going to have something else at the other end. And so if we look at this diagram at the bottom, we can see where the mouth is forming, so that's where the food's going to go in. Just like you and me, they have a one-way digestive tract, and so you have the digestive system, and the food's cruising on down the intestine, and eventually the food goes out the anus, as food should. However, and so they have a respiratory system, digestive system, an endocrine system, nervous system, those sorts of things that we have. And kind of in the same sorts of places as we have ours, except the respiratory system. Now, as we know in us, almost all organisms, the respiratory system is associated with the mouth. You breathe with your mouth. Not cucumbers, no. A sea cucumber's respiratory system, referred to as the respiratory tree, actually enters and exits at the anus. Sea cucumbers breathe through their butts. And we're going to have this come back to haunt us in our dreams later. Now, when I talk about these animals rotting in the sea and regenerating in the sea, I'm going to kind of give it away here. There are points in time where we can look at one of these sea cucumbers, and if we invite it to open itself up, say we take an animal in the summer, we're going to see the full set of internal organs. Everything in there just jam-packed. If we were to take an animal during the winter, like if we went there today and opened it up, we see hardly anything at all. What these animals are doing during the winter time is entering a state of hibernation. All of their internal organs are disappearing. What in the world's going on here? Well, this is the puzzle that we're going to try to solve. And again, I can't do this by myself. I need a lot of help. And fortunately, we have a graduate program at Walla, and so I've had a variety of graduate students who have been involved in just the past few years. These are students who have graduated within just the past 1 to 3 years, doing a variety of things now. Amy Tan is in a PhD program down at Texas A&M studying regeneration in fish. Down here, Robin Reeve is in a PhD program at Washington State University studying regeneration in frogs. Hannah Starcevich is in an infectious disease PhD program at University of California, Davis. Chanel is working in a biology lab at the University of Idaho. Julia's now in a pharmacy program at University of Washington. Chris Parker is in a MD program in Oregon. They've gone on to do great things, and so we'll talk about some of their research. We're also going to try to talk about a little bit of the research of my current students. They're back in the lab right now while I'm here playing, having a good time, eating Peruvian food. I'd never eaten Peruvian food before. Go to that restaurant. One student, Flora Gibbs, just sent me a text about a half hour ago with a couple of exclamation marks showing me a beautiful slide that she's preparing looking at the neurotransmitters in the nervous system of these animals, and she finally got it to work. We can now track epinephrine and norepinephrine and serotonin. I'm really excited. Chris Lindsay is studying the bacteria which naturally live in these animals and how that changes. And then Faith Honeycutt, who I think there's a few people in here who may know Faith, she is looking at telomeres, and we're going to be looking at those in just a little bit. Well, let me first talk a bit about this whole process of science. Word we throw around a lot. Research, word we throw around a lot. What are these things all about? Well, for me, science, as you can see, is simply observing something in nature that really interests you. And after you've observed it, you go, that's kind of odd, or that's pretty cool, that's weird. What in the world is going on? I want to know more. Well, trying to figure out what's going on, you come up with some natural explanations to explain these natural phenomenon. You don't know whether your explanations are right or not, but let's try to figure it out. And so you do some experiments. Well, oftentimes we don't get it right the first time. It's part of the process. Science involves a lot of patience and trying things over and over again. But eventually, we're going to come up with some explanations that we can examine that might explain the phenomenon that we're really interested in. And not only are we interested in this one little thing, but we want to try to see some broader implications of our findings. And so this is the approach that I'm going to be taking now. We're going to study this organism called Parasticopus californicus, the California sea cucumber. It occurs along the entire west coast of the United States and also extends up into Canada and down into Mexico. And so that's what we're examining here. Some of you are from down the LA area and Loma Linda and Redlands and those kinds of places. There's some people. Anybody here from Alaska? No. Well, if you were from Alaska, you'd be way up here, and there's some people from Portland. And so we can see the range of this species, all the way from very cold Canada to somewhat warm Baja California. Where I study the animals are right smack in the middle where we have our marine lab. Well, we start with making some observations of these animals. And for several years, we've been examining just the overall size of these animals. And we've observed a very strange thing. If we grab some of these animals in like May or July, summertime, we see that they're really big. As you can see, around 1,500 grams, 1.5 kilograms. If we were to go there today and grab these same exact animals, what we would now see is they weigh maybe 600 or 700 grams. They're losing up to 2/3 of their body mass, and they do this every single year. We're not sure how long sea cucumbers live. It's estimated a minimum of 30 years. And so every year, they're going through the cycle of losing 2/3 of their body mass and gaining it back again. Imagine if you did that. Well, we want to know what's going on here. Well, when we examine these animals, again during the summertime, we see they have all their internal organs. And that's why they weigh so much. But then in the wintertime, all their organs are now gone. And so this is an animal which is opened up during the month of January. There's no digestive system, no respiratory system, no reproductive system, no nervous system, no nothing inside of this animal. That is absolutely bizarre. And they do this every year. How in the world can these animals lose all their internal organs every year and grow them back? We see some interesting things. If we go in the summer, these guys are crawling across the ocean floor, moving, and they're eating and reproducing and all that kind of good stuff. Around November, they stop. And while they're stopped, their organs disappear. And they stay sitting there unmoving until about March. March, over a 2-week period, the organs regrow and they just start moving again. How cool is that? Well, what's going on? Obviously, there's some kind of atrophy or degeneration which is occurring on a seasonal basis. We go from July, the summer, when they have all their organs, now to November, the fall, and into the winter, where they have absolutely no organs. And then, come March, their organs grow back again, they get their full organs, and they start this cycle over and over and over again for the entire life of the animal, potentially decades. Well, there's several questions, big questions. Why in the world do they do this? I'll tell you right now, we have no clue. How do they do this? What are the mechanisms that are involved? We have a few pieces of the puzzle that might go to explain that. First of all, why do they do it? Why lose all your guts just to grow them back again? We don't know. We have some hypotheses, some reasons. Maybe there's an energetic savings, but there's a huge energetic expense to grow them back. Maybe there's simply no food, and so there's no food, why have a digestive system? But there's still the huge cost of losing it and growing it back. Do cold temperatures somehow cause the loss of these organisms? One hypothesis, maybe there's internal parasites. I talked to a parasite lover today. And maybe the parasites need to be gotten rid of. The only way to do that, well, get rid of all the organs too. Maybe there's some osmoregulatory issues which are happening during the winter. All hypotheses. I have a couple new students want to start tackling this. We have no clue why they do this. We have a few answers as to how they lose and regrow their organs. And so these are going to be some of the experiments that I am going to talk about now. So how do the organs disappear? Where do they go? They kind of store them in a suitcase or something. How do they reappear? How long does that process take? They're not eating. If they're not eating, they're not taking in proteins, nucleic acids, and fats, and carbohydrates to regrow things. So how do they make new organs? Well, let's first look at one of those first things. How do these organs disappear? Well, if you have an organ, let's take your heart. Keep it though. Let's have an organ. If it's going to disappear, that means the cells have to disappear, which means the cells have to die. How do cells die? Well, they just die, don't they? There's actually a couple main ways cells can die. One is just to kind of rot away. It's what we call necrosis, where due to some loss of energy or some catastrophic event, everything just starts to die. That's what mainly happens in us from time to time. It's very unregulated. It's very catastrophic. But it's very easy to tell a cell that has died through necrosis because the cell all of a sudden gets bigger. It swells. All the organelles, the mitochondria, Golgi, they all swell and get bigger. And then all of a sudden, the plasma membrane just ruptures and basically pops like a balloon. Easy to tell. However, there is another way that cells that can die, a very controlled way, a purposeful way, which is called apoptosis. It's very common during development in us. It's why you don't have webbing between your fingers and toes. You did at one stage when you were inside your mommy, but through apoptosis, all those cells that were there died in a very controlled fashion. And so in apoptosis, just the opposite happens. The cell actually shrinks, and we get these little fragments which form. They're kind of called blebs. The cell is blebbing. And we get the nucleus, which starts to fragment. Merely by looking at cells, we can tell which one of these processes is happening. And so we wanted to figure out how in the world are these cells disappearing in these sea cucumbers. Not only can we tell by a microscope, we can tell by looking at the processes which are happening involving enzymes. Now you're probably aware of course you know about enzymes. They're biological catalysts, catalyze various chemical reactions. There are a series of enzymes which are called caspases, and these are involved in apoptosis. They're given numbers. So there's caspase 1, 2, 3, 4. How original. Several of them are what we call initiator caspases. So 2, 8, 9, and 10. They get the process going, but don't do the actual killing. The ones that get the killing done, I love the names, executioner caspases, 3, 6, and 7. And so we're going to see that there are times where a signal comes from the outside in what's called the death receptor. I just thought of my next Halloween costume, all right? The death receptor. And so we have initiator caspase 8, for example. But then we can have internal processes which start to mess up. And this can also then cause certain of these initiator caspases, caspase 9, to get the whole process rolling. Well, so I had a student who was examining these caspases to see what changes might occur, and it was looking at enzyme activity. What we're examining in this particular figure are two of these initiator caspases, 8 and 9, and two of these executioner caspases, 3 and 7. And we're going to examine them during these 3 times of the year. Green, the summer, when they got all their guts, everything's there. The fall, when they're disappearing, all right, atrophy is occurring, things are dying. And then in the spring, red, when things are growing back again. Well, not too much of a surprise, we saw that at least one of the initiator caspases, an increased activity during the fall. Degeneration is leading to the increased activity of some initiation. And we also saw for both of the executioner enzymes caspases that we examined, an increase in activity during degeneration. So that tells us that apoptosis is probably one of the main things causing cell death, is controlled cell death during the actual organ loss. What confused us, however, was that during regeneration, we saw that at least one of the initiator caspases and one of the executioner caspases were active. That during regeneration, apoptosis can be occurring. That was like, wait a minute, during regeneration we're trying to grow things. Why do we want death? Same reason why maybe we had the webbing disappear from between our fingers and we were growing. Normal developmental growth processes does involve certain death processes as well. So during this degeneration, we have caspase 8, which is associated with the death receptor, which is activated. During regeneration, we have the initiator caspase 9, which is activated, an internal process. Bottom line, apoptosis appears to be responsible for the disappearance of these organs. So as we go from summer to winter, as we go from lots of guts to no guts, lots of apoptosis is causing the organs, the cells, to die into the individual molecules. Those individual molecules are then lost from the animal out of the anus. So the animal's losing all the molecules out of its rear end. Doesn't store these molecules at all. Gone. Weight loss. Hibernation occurs. What about the opposite? If it's now lost all of these molecules, where is it getting molecules from to now regrow the organs? Got to have molecules if you're going to regrow organs. What's the source of the biosynthetic material? Where are the proteins, nucleic acids, carbohydrates, lipids? Where are they coming from? Maybe stored. We might think, well, maybe they store fat, and that's what they're using. But they're not. Because as we said, these animals during the wintertime, they've lost all their organs. They've lost a ton of mass. There's no storage at all which is occurring. So it's not stored material. Well, maybe they're just feeding. If you eat a lot, you're going to gain organic molecules, but during the winter, these animals don't even move. In fact, they don't even have a digestive system. So they're not feeding. How are you going to get organic molecules if you're not storing them and if you're not feeding them? Well, that's what we want to figure out. And so we thought on a whim to look at what's referred to as dissolved organic material. Now, if you do, who all are scuba divers in here? A. Who wants to be a scuba diver? Yeah, forget it, too late. When you scuba dive in certain waters, especially up off the western coast of the U.S., the water is pretty murky. And so we see all these kind of dots around all over. The murk is, there's a lot of stuff in the water. All right, a lot of plankton, a lot of little animals, little plants. There's also a lot of dissolved organic molecules, a lot of molecules. And a lot of those molecules happen to be amino acids. And so if we look at a diagram here, this is a big old sea cucumber on the bottom. let's imagine that there are a whole bunch of amino acids just floating around in the water. Maybe these amino acids can be absorbed by the animal. The animal can then put the amino acids together to make proteins and make everything else that it happens to need. Well, how do you know if an amino acid, if this individual amino acid has gone into the animal? How in the world do you figure that out? Well, what we do is we put little labels on these amino acids. We use stable isotopes. And so we can make our own amino acids that rather than carbon 12, they now have carbon 13. Or rather than nitrogen 14, they now have nitrogen 15. We can follow individual molecules and see where they go. And so that's able to tell us then, are these particular molecules then being absorbed by the animal, maybe right through the skin? And that was our hypothesis, that they're absorbed through the skin. And so we couldn't do this out in the ocean. We had to bring the animals into the lab. And so here's 4 of the animals during the summer where we've thrown in these labeled amino acids into these chambers and see how many are taken up by the animal. Well, in this particular figure, we're looking at basically how many of these amino acids have been taken up. 0, no surprise, means that none have been taken up. We thought lots of them would be taken up by the skin. When we look at the epidermis for the 3 different seasons, summer, winter, and spring, none at all. If we look at the digestive tract, well, there really isn't one, so there's none taken up there. If we look at the tentacles on the front of the animals, a little bit. If we look at the tube feet, a little bit. But obviously, where we see most of it, totally caught us off guard. We thought, well, let's just measure anus tissue because there is anus tissue and it's fun saying anus tissue. So we measured anus tissue. That's where it was. That's where these amino acids were being taken up. And so it hit us like a ton of poop. Sea cucumbers are eating with their anuses. Not only breathing, eating with their anuses. So when these animals are sitting there, no digestive system, no eating with their mouth, no moving, individual amino acids are being taken up through the anal tissue. These amino acids are then being incorporated into proteins, converted into lipids, carbohydrates, new tissues. You get organs which form in 2 weeks. That's pretty cool. So we're solving some of the issues here as to what's going on, but there's got to be a lot of control mechanisms with these animals. How are things being turned on and turned off? How does the animal know when to do this and when to do that? Well, one thing we can look at are changes in gene regulation. There's got to be certain genes turned on now and then turned off then, and other ones being turned on then and turned off now. And so we decided, let's study some of these genes and see if we can determine when they're being turned on and turned off. There are probably hundreds of genes involved with this process, and we're slowly working on figuring those out. But first of all, we decided to look at a couple that we'll talk about in a second. Now, this kind of diagram is something that you better recognize, that this purple here is the nucleus. And of course, as you know, double-stranded DNA in eukaryotic organisms containing your genes. We know that during the process of transcription, we can form a molecule of messenger RNA. The messenger RNA, as you better know, then leaves the nucleus, and outside in the cytoplasm, it can then combine with a ribosome. A for him. Wait, was that you? Are you a diver too? No, it's just an A. Okay, I was going to give you an A+. And then we get the formation of the protein. Now we know it's the protein that does the work. The protein is what does the work. But what we were going to examine was how much messenger RNA is present, because that tells us whether the gene is being turned on or turned off. So which messenger RNA, which proteins were we interested in? So far, we have a really good idea what's happening with 2 of them. One is called survivin. What a cool name. Now, we have survivin, I have it, you have it, okay? And it's highly involved with cell proliferation and differentiation, especially during development. It's also seen in wounds as they're healing. You see the survivin present. Any cells which are rapidly dividing, even cancer cells. And so that's why people are very interested in studying survivin, where it's found naturally and also found unnaturally. And as you can see there, it can inhibit apoptosis. Another one is called mortalin. Mortalin can also inhibit apoptosis. Mortalin is usually present when there's some kind of huge stress inside of the cell. And as you can see in that last point, it's been implicated in certain neurodegenerative diseases. Well, these animals have a nervous system which degenerates and regenerates. Maybe mortalin is involved here. And so, while it's the proteins that do the function, we were interested in the messenger RNA. Are there changes in how much transcription occurs? Are there changes in how many copies of the messenger RNA there are? And so that's what we decided to examine. You do that using a process called qPCR. Maybe you've had the chance to run electrophoresis gels before, where you get these cute, pretty bands. And by doing all this stuff, we were able to determine how many copies of messenger RNA there were. Now, we're looking, for example, in this first figure at the survivin messenger RNA and how much it changes from a control level. Our control level is the green during the summer. And if we look at the intestine and the muscle and the respiratory tree, we see that between the summer and the fall, no change. But during the regeneration, huge increases that are occurring in how much messenger RNA for survivin there is. We saw the same thing with mortalin. Big increase in the intestine, lots of regeneration, big increase in the respiratory tree, lots of regeneration. And so this is telling us that at least these two genes are highly turned on during the regeneration process. So you have regeneration, you have these tissues which are growing. They're growing from nothing, which means you're probably starting from one cell or very few cells. Well, if you're going to go from one cell or a few cells to an entire tissue, you got one cell's got to become 2, and the 2, 4, and the 4, 8, and the 8, 16, and on and on and on. A lot of cell division. And then during the winter time, they start dying off. And then the following spring, they divide and divide and divide and divide. And then they die off. I'm having lots of fun. And then they do this again. Is there a limit to how many times cells can divide? Can a group of cells divide forever? Are those cells immortal? And the answer is no. Mainly, in most organisms, such as us, cells have a limit as to how many times they can divide. What causes that limit are these structures on the end of the chromosomes called telomeres. If we look at this X down at the bottom, representing a chromosome, on the very ends of the chromosomes are these sequences of DNA which are called telomeres. Every time a cell divides, the telomeres get shorter and shorter and shorter. So at first, you have a big telomere, and then it gets a little shorter, a little shorter, a little shorter, and at some point in time, it completely disappears. No more cell division can occur. So another diagram, the telomeres get shorter and shorter and shorter until eventually they disappear, no more cell division. Well, that's what happens in us, all right? My telomeres are probably shorter than yours are. My cells are going to stop dividing in about 2 minutes, and I'll keel over dead. And you get to continue the talk. Just push the button. That's all I do. Okay, thank you. All right? Is that what happens in these sea cucumbers? These animals are regenerating and regenerating every year, every year, every year, every... Are they immortal? Or are they like me, where they can only regenerate a certain number of times? Do we see the same process occurring in the sea cucumbers where the telomeres disappear? No clue. This is a study we're doing right now. My student Faith Honeycutt's working on this as we speak. We've gotten a little bit of data, including some in just the past few weeks. And so what I'm sharing with you now is extremely preliminary, all right? But we do qPCR in this. And here's what we have found. If we look at the telomere length in an animal, well, let's look at the intestine and respiratory trees. These are the tissues which are regenerating over and over again. And what our preliminary data is showing us is that during the actual regeneration, telomere length is doubling to tripling. Telomeres are not getting shorter and shorter and shorter. They're getting longer and longer and longer. Absolutely opposite of what happens in us. What in the world? How do you make longer telomeres? What we're also examining then is an enzyme. The enzyme is called telomerase. It's an enzyme which can cause the telomeres to grow. And again, these are very preliminary data, but if we look at the intestine and the respiratory tree, and the red is the winter, we see that the activity of the enzyme goes up 4 to 5-fold. Whoa! Well, again, that's the protein, what does the work. That's the protein. How about the gene that turns it on? What about the number of copies of messenger RNA? Is the telomerase gene turned on, which leads to the telomerase, which causes an increase in length? And if we again look at intestine and respiratory tree, we see that the number of copies of messenger RNA are going up 4 to 6-fold. These guys are doing absolutely the opposite of what normally happens. Rather than telomeres getting shorter and shorter and shorter, and so eventually the cells can no longer duplicate, can no longer divide, telomeres are getting longer. Does that mean that these animals can live forever? We don't know. We don't even know how long these guys live. How can we not know how long a particular organism lives? Something that we want to study. So, from our puzzle, we do have a few answers. We have a good idea that the loss of organs is probably caused by apoptosis, a very controlled process, not just tissue death due to necrosis. We know that organ regain, the regrowth of these organs, the regeneration, is due to certain genes at least being turned on, at least the 2 genes mortalin and survivin. But there's going to be more that are involved. We know these animals are not eating, they're not using stored material, but they're absorbing dissolved organic material that's in the surrounding seawater through their rear ends, and from those individual molecules, remaking all their organs in about a 2-week period. And we know that contrary to what typically happens, telomeres are actually increasing in length. Apparently, we got a lot more work to do on that thing. Absolutely bizarre. But this leaves us with lots and lots of questions, tons of questions. A huge one, why in the world do these animals do this? What are the ecological pressures that cause these animals to go through this cycle every single year? Why lose all your guts to grow them back again? We don't know. How can we not know? We don't know why we don't know. What tells them to do this? What tells an animal, okay, dude, stop where you are and lose your guts? Changes in photoperiod, food availability. Is there an internal clock, regardless of what the external environment is doing? We don't know the answer to that either. You know, who does this process? We're studying animals in the middle of the range. What about the animals way up in Alaska? What about those animals in California or down in Baja? Animals in the whole range doing it? No clue. Nobody's looked. What about other species? None that so far that we've examined do this except for Parasticopus californicus. How often can they do this? We know they do it every year. Could they do it multiple times a year? Can they do it for 50 years, 100 years? Don't know. My telomeres are getting shorter. I won't find out if they can do it 100 years. Why can't I do it? I'm a human. I'm advanced. I'm the most complex thing in nature. Really? I can't do this. I can't lose my guts and grow them back. Or can I? I feel them going now. How cool would it be if I have a bad liver to just let it dissolve away and whoop, maybe not eat through my rear end, but somehow cause this new organ to regrow? I'm advanced. Sea cucumbers are simple. They can regrow all the organs. If I have an infected kidney, if I have a bad heart, why can't I regrow an entire organ like a supposedly simple animal can? And so that's sort of a larger picture. I love sea cucumbers. I love studying degeneration and dormancy. But for me, I want to know things from an ecological perspective, an environmental perspective, behavioral perspective, a molecular perspective. And what are the larger implications of these things? We have figured out a few pieces of the puzzle. There are many, many more to go. So if anyone's interested in getting a master's degree, interested in this kind of research, interested in any of this stuff, feel free to ask questions. I am happy to take your questions. Thank you very much. Come on, it's got to be questions. You can't leave yet. They told me that I get to dismiss you. You're not being dismissed until there are 18,000 questions. Okay, or one. Go ahead. What got me interested in this? This is a great question. I'm going to go back to my undergraduate years, way back. I was once like you. And during my undergraduate years, I got involved in the research project with, get this, sleep. Oh, it was awesome. We got to hook people up with EEGs and watch EEG patterns and things. And we did it on some wild animals as well. And from the sleep, it was like, man, these animals are shutting down. And so I got interested in how animals can shut themselves down. And so when I went to grad school, I studied hibernation in little tiny mice called deer mice. Oh, that was great, watching little deer mice shut down. Little deer mice get a virus called hantavirus, which at that time had a 70% mortality in humans. I was okay with freshmen using them, but no, I wasn't. All right? And it was like, so for 2 years, I still worked with them, but we were wearing biohazard suits, respirators, and I got to thinking, it is only a matter of time before one of us gets this. It is not worth it. What am I going to study now? And it was a student who brought it to my attention. She'd read a paper from the 80s that suggested that these animals might go dormant. Boom! That's how I got involved in these animals. Woo! Thank you. Other questions? I thought I saw a hand somewhere. Yes?
Is the regeneration based only on temperature, or are there other environmental factors?
We have no clue what the environmental factors are that cause this to occur. And so one thing that we need to do are controlled lab studies. So if you kept the temperature constant, would they still lose their guts? We don't know. If you kept photoperiod constant, food availability constant, we have no clue what the environmental trigger or triggers are. Many animals where we know the triggers, it's usually temperature or photoperiod. Those are the first things that I would check with. Those would be my initial hypotheses. Those would be the first things to check. Good question. 2 more questions here. You're not going anywhere. So what I'm going to do is I'm just going to point at random people, and you have to ask a question. How's that for putting you on the spot? That'd be cruel. But it'd be so much fun. Okay, yes?
What other animals would you like to study?
What other animals? I love everything in the ocean. I love marine biology. There is so much down there. Those of you who have scuba divers, you know it's an entirely new and different world that all you land-dwelling people never get to see. And it's just phenomenal. I like things that live in muck. Why? I don't know. So worms really kind of interest me as well. Tardigrades, those little microscopic water bears, those are really cool too, and I like to study tardigrades. Yeah, and then one back there. Go ahead.
I was just going to say, what's some other holothurian behavior that you find fascinating?
Other holothurian behavior? Well, breathing through your butt isn't enough. There in the Antarctic, there is a sea cucumber called the vampire sea cucumber. It has these little wings and this spear, and it will swim around and will stab and spear fish. That's cool. I thought I saw a hand back here. Yes? Do any other echinoderms exhibit this sort of behavior? There are other, if you take a sea star and you hack off an arm, it'll regenerate the arm. There is one species of sea cucumber that if you cut the entire animal in half, both halves will grow into a whole organism. But I don't know of any other organism that on a yearly basis loses all of its guts as part of a yearly cycle and then grows them back. There are some sea cucumbers that if a predator comes after them, They'll puke up some of their internal organs. The predator goes, yum, eats the guts, and the cucumber can then kind of slither away. But this process, I don't know of any other organism. Yes?
Do you think that some scientists can use this as an evidence that organs can start existing from almost nothing? Or from like small?
Could a scientist use this to show that organs could exist from almost nothing? That is what's happening here. Is there really absolutely nothing? There's probably little teeny nubbins of digestive system, maybe a few hundred cells, and so it regenerates from that. That is happening. Can we do that? We're starting to figure out how to grow new organs, in the lab. But in humans or any mammal, there's been no situation where you can get an entire new organ from just a handful of cells. Not yet. Fascinating. Yes?
So during the winter, when they're lacking all of their organs, are they just completely hibernating, or like how are they functioning, or like eating or drinking without?
Yeah, what are these animals doing during the winter? When they stop crawling around November, we've measured their metabolic rate, how much oxygen they're using, and it decreases about 90%. So a lot. And so they're still alive, even though there are no organs. They're still absorbing oxygen somehow, even though they have no respiratory system. We're trying to figure out how they fight off diseases if they have no immune function, or was one thing that we're trying to look at. And they sit and they do nothing. So I can find, animal Fred, and in November, mark where animal Fred is. I can go back in January, just did this a few weeks ago. Water is cold, and animal Fred's still sitting there, hadn't moved at all. Still facing the same direction, bored to tears. Yes? Are there any, good question, are there any predators? The number one predator of this species? Humans. I know, people, they taste nasty, don't try, trust me. No, don't trust me, try. Okay, it's soy, so you vegetarians, it's okay. But, what was I saying? Predators. Human, shiny, humans. And so, there's a huge fishery for these animals. And usually about August and September, you'll see lots of boats going out, and they take up hundreds of tons, ship them. Most of them get shipped over to Hong Kong because they're a delicacy in certain countries over there. In terms of natural predators, I have never seen one of these with a bite. I have never seen anything attack these. I don't know of any natural. I take that back. There's this big old starfish called the sunflower star. And you will see sunflower stars go after these animals, but if a sunflower star touches one of these sea cucumber, the sea cucumber basically runs away. Other than that, I've never seen a half-eaten cuc or one with a bite or a predator with a cucumber hanging out of its mouth. Even in the wintertime. And they're not even moving. It's like fair game, free food. Cuc sandwich. Yes? Parasite. Oh, man. We have, if you look on the outside of these animals, I should have brought some pictures. There's these little isopods that live on the outside. We don't know if it's acting as a parasite, a symbiont, mutualistic sort of thing. And if you look inside of them, in late summer, there's lots of little squiggly worms. But in the spring, we don't see those little squiggly worms. And that's why one hypothesis, not at all tested, maybe the only way to get rid of the parasites is to get rid of all your guts. And then you grow your guts, grow your guts, grow your guts back again. Okay. One last question before you go study OChem or, okay, two more. Here, then here.
Do their anuses always stay open like that?
Oh, what a great question. Thank you. Their anuses do not always stay open. Here's the deal. When they have their full guts and their respiratory trees, you'll see the anus closed. And then all of a sudden it opens and closes, opens and closes. What's going on is it's inhaling some water and then closing and slurping in water, slurp in, until its respiratory system gets filled with water. And then all of a sudden, the anus opens huge, and it flutters, and you see all this water come pouring out of the animal. So open and close, flutter, and all the water comes out. You got to be careful when you get too close, because sometimes the poop comes out too, and it kind of shoots out like a little BB gun. Oh, not this species. There are certain species of cucumbers where there's a little fish that lives in their anus. What a nice house. And then there was one more. I thought, there it is. That do what? Have we found any not going through the cycle? We do see some slight fluctuations in the dates. Some years, if we go in October, they're all already hibernating. Other times in October, none of them are hibernating. But we have never found one in, say, December, January, February that isn't doing this. So we've never found one that isn't doing this. There could be some that we simply haven't found, because we're not going to go like chop up 1 million. Okay, last question probably. Do they have any defense mechanisms other than running away? Not really. There are a few anecdotal scientific studies that suggest that they may contain some noxious chemical that could drive away predators. I've eaten them, I've licked them, and nothing's happened to me. I'm fine. But what? Somebody once taught me you can't truly study an organism until you eat it. So anyway, all right. Who are the keratologists and the bronchiologists in here? Yeah, those are the things that you can eat. Okay. So anyway, I don't know of anything, but they don't get eaten. Part of it may be, if you look at the nutritional value of one of these things, they're a bag of water with some guts full of poo. And so there's just not a lot of in them for something. Thank you. That's exactly right. Okay. Thank you very much. I'm going to be around tomorrow.